Hello. In this poster presentation, I want to describe an approach how to efficiently analyze genome data at the network level. It's called network propagation of multi-omics data for the identification. So in the area of high throughput sequencing, it has been proposed to measure, for example, in personalized medicine of cancer, a lot of data at different levels of molecular information, for example, gene expression or protein expression, and to bring this data into context by mapping the different data onto networks of interacting proteins, and then to derive, for example, thematic subnetworks, which better describe the mechanisms than each single data set. Another motivation of this is that often signals have a heavy tail distribution where, for example, mutations in this case, mutation has a high frequency with respect to some genes, but very low frequencies with respect to other genes. And these genes or these mutation patterns can be best brought into context through mapping them onto networks. Now the mathematical way how to do this is with network propagation. Network propagation builds on the guild by association paradigm, which says that genes that underlie similar phenotypes tend to interact with each other, so that from molecular interaction networks we can basically derive function. And network propagation is a stochastic process which starts by initializing some nodes by experimental values, then diffusing the weights of these proteins through the neighbors in the network, and then do that through time and compute a steady state distribution or convergence state. And from this convergence state, basically derive the subgraphs, we call them network modules, where most of the weights have been assigned to. This has the benefit that the nodes get re-ranked compared to the initial situation, that effects in the known genes can be propagated to identify network modules and thus better describe the mechanisms underlying the phenotype of interest. And the effects in known genes can be propagated basically to derive novel biomarkers, in the sense that we get weights for nodes which have been unweighted at the initial step of the approach. Now, for the approach, we need a scaffold of molecular interactions, and my group has developed since years the consensus PathDB, where we basically collected and integrated molecular interactions from 32 public resources. We have different interaction types, and among the interactions, we also have a large amount of protein interactions integrated from different resources, around 600,000, which we use as a scaffold for network propagation. The PPI network content of the consensus PathDB, as I said, is around 600,000. However, we made a confidence assessment of the protein-protein interactions based on different measures, and we can basically assign a high confidence interaction network, which consists of something like 140,000 different interactions, where we are very sure that the interactions are not false positives, but rather true. So we stick to this part of the consensus SparseDB in the following. What we also see is that we have a small world network, that most protein pairs have distance below 5, and we have a few hubs and many genes with a low degree. Now, the PPI networks have been benchmarked by an independent group, and for example, on their ability to recover disease genes, and the consensus SparseDB has performed as one of the best PPI networks that you can get nowadays. The second ingredient of the approach is basically the network propagation algorithm. Here we developed the tool TwoNet Core, which is a two-step approach. First step, we derive a re-ranking by using the core of the network as a node characteristic rather than the degree, which is a part of the most other available network propagation algorithms, but we observed that the core is particularly suited to cope with the bias of PPI networks and to identify influential nodes. And the second step is module identification. So from the re-ranked graph, we identify modules where much of the action happens. This approach has been published as well, and we can discuss it at the poster. We applied this approach to two applications. So the first application is a toxicity project where we were interested in toxicity mechanisms from anthracyclines, and we derived an integrated anthracycline graph from the network propagation, which can be subdivided into a mitochondrial part, the sarcomere part, and the extracellular matrix part. This has also been published. The markers that we get, or what we observed, is that many of the sarcomere genes are already cardiomyopathy disease genes, and that from the network we can derive translational biomarkers which are expressed in patient biopsies and are correlating with the clinical parameter, within that case is the left ventricular ejection fraction. In another project, we looked for differences in pathogen infection responses in resistant and susceptible mouse strains. In this case, with the pathogen Leishmania major, which is a disease which is common in Mediterranean area. And what we see is we can derive different network modules for the susceptible and the resistant strain. We see that the immune response, for example, is much higher in the resistant strain, and we derive different metabolic pathways, which we can also discuss at the poster. To summarize, omics data agree rather on the level of pathway networks and biological functions than on the level of molecular entities. We developed an integration concept that can explore the different omics data sets, and that is based on network propagation in protein interaction networks. We computed toxicity network modules and found that we can assess novel biomarkers that are clinically translatable. We use that for network analysis for inferring different responses to pathogen related to susceptibility and resistance. These are the acknowledgements, and thank you for your attention.